All right. Um, so we ended up just doing some sort of basic diagrams of thyroid pathways. But for the thyroid in particular, we're going to talk a little bit more detail about some of the cell signaling. We could do this for all of them, but we don't really have time, so you're lucky. I'm not going to require you to know more about each of them. But this one's kind of an interesting one because it uses multiple pathways. It's neither lipophobic or lipophilic. It's kind of both. So it comes in a sort of different fashion than some of these other hormones. Just going to erase my weird line over here. So let's go back to that. So we know from looking under the microscope that we saw this already, where right here you have cells that are follicular cells, as in cells that make up the follicles. And then inside of the follicle is colloid. Okay, that fluidy gel-like stuff that's on the inside. And both of those things together make up the entire follicle, which is that large circular structure, not large as in, baseball sized, but large as in on the microscope you can see it pretty well. And that follicle is responsible for making T3, T4, which is the hormone that's responsible for increasing and regulating metabolism. So it's going to start up some of those metabolic processes. Okay, right here we have parafollicular cells, as in cells that are parallel to the follicle. So parafollicular cells also secrete hormones, where the hormone that they're responsible for is calcitonin, which deals with calcium homeostasis. Calcium homeostasis. And we will get to calcium homeostasis, just not quite yet. Now, when we secrete from this thyroid follicle T3, T4, what causes the follicle to start secreting? Do you remember? What causes it? TSH, thyroid stimulating hormone. And where does TSH come from, Kelsey? It's the anterior pituitary. Good. So the anterior pituitary is going to secrete TSH, which is going to tell the thyroid follicles to start secreting. What tells the anterior pituitary to start secreting TSH? The hypothalamus, which releases what? A releasing hormone. Thalamus. In this case, the releasing hormone is called TRH, or thyroid releasing hormone, right? Which makes sense. So we have that pathway that's still here, and now we're just going to look at what's going on right deep in here. So if we take this cell that's just gotten that signal that now it's time to make some T3, T4, that's going to be this cell. So this is a follicular cell. Over here on this side is going to be interstitial fluid. We know there's actually going to be a bunch of cells going up this way and going down that way. They're all attached to each other. And then on the inside, this is going to be the colloid. Actually going to move this up here a little bit and get it out of the way because we're going to need that space. So this side is going to be the colloid. All right, so we've gotten a cellular signal that says, okay, it's time to start making some T3, T4, and that signal is going to start a reaction that happens in the nucleus. So if it's going to be coming from the nucleus, what do you think, what kind of structure are we probably going to make? Yeah, we're going to end up making a protein. So we're going to make mRNA, and it's going to head to that rough ER where we've got ribosomes, right? So rough ER and ribosomes on the rough ER. And the rough ER is going to make a precursor hormone called thyroglobin. Thyroglobin, which is a precursor. Okay. The thyroglobin is going to go to the Golgi. Where it's going to be put into a vesicle. With digestive enzymes, meaning they can break things off. Okay. I'm just going to add my little thyroglobin in here. Okay, and then that thyroglobin is going to be exocytosed into the colloid. So thyroglobin, and it's no longer a precursor. It's still got to do some modification, but it's thyroglobin straight up. Oops, to colloid. Okay, so we've just got this thyroglobin that's going to hang out over here. All right. At the same time as all of that is happening, we need another additional element that we're going to add to that, and that is iodine. Iodine. So iodine is just going to diffuse straight through that follicular cell into the colloid. Iodine is just going to diffuse. Okay, when it diffuses, it's going to come and add to this thyroglobin. Okay. So now we've got this thing, but then it also has this other little iodine on it or something like that. And I love my precise language there. So then when TSH, that thyroid stimulating hormone, is present, then we're going to take this and it's going to go through endocytosis back into the cell. Okay. So it's going to head back in here, and now we've got this little vesicle. It's got some protein parts, some iodine part, and the iodine part is nonpolar. It's diffused, so we know it's nonpolar. Okay. Now if I'm just going to do this as a little note. So if there's no iodine present because of diet or for other reasons, if there's no iodine present, then we can't release thyroglobin. This is one of the symptoms of if you aren't making enough T3, T4, you end up not being able to release thyroglobin, and it leads to an enlarged thyroid gland. Because all of those follicles get enlarged because they're making thyroglobin. It keeps filling up with thyroglobin, but it can't be back released because there's no iodine to attach. That enlarged thyroid gland is called a goiter. I'll show you a picture of one in just a few minutes, hopefully. Okay. So if there's no iodine, we can't release that thyroglobin at all. Okay. So the same thing is going to happen over here where we have some digestive enzymes added. So this is a vesicle with digestive enzymes from the Golgi. That's Golgi's job. It's going to do some packaging. It's going to do some modification. And at this point, we're going to modify thyroglobin plus iodine into T3, which in turn is going to be modified into T4. T4 is the most usable version of this molecule in humans. We can have different levels of T3 and T4 within the body at any time. You don't have to modify all of the T3 at this point. We can also secrete it. It gets modified in other cells. So then this is going to travel this way, and it's going to be released. That T3, T4 is going to be released here through exocytosis again, because we've got some polar, some non-polar sections, and if there's any polar sections, then we can't just diffuse. Okay. And now, because we've got this T3, T4 with some polar regions, some non-polar regions, well, it couldn't diffuse out because of the polar regions, but it also can't make it through the bloodstream because of the non-polar regions, so it needs a carrier. So there's going to be a carrier protein to move through blood to the tissues. And then we're going to release that carrier. Off the carrier. And it's now going to bind to a receptor at the target. And that, in turn, is going to increase transcription and translation of specific genes. Which in turn is going to increase metabolism. One of the biggest products of this increase in metabolism is an increase in heat. So if you have someone that has an incredibly high metabolism because they have too much thyroid hormones, you might have them be really hot. That's one of the symptoms of that. And that's the end, right? So note that we have a lipophilic, philic/lipophobic, depending on what stage we're in. Where the lipophilic in particular is notable because not all are hormones. In this case, we made it lipophilic by adding iodine to that first original protein molecule, and so that changed it so that over here it was able to just move into the cell by diffusion. So it's a really unique molecule that doesn't really follow the rules that I just taught you. Isn't that nice? Any questions about that? Make sense? All right.